Hi, in this module we're going to be talking about fMRI data structure. So this is going to be important when we get into the data analysis and the statistical analysis of fMRI data. We have to try to understand how the fMRI data is structured and know a little bit about the nomenclature. So standard fMRI experiment gives rise to massive amounts of data. So we talked about that in the last module. And most standard fMRI studies consist of both of structural and functional data. And here we discuss the structure of the data and some general terminology that's associated with it. We're also going to provide a brief overview of some of the characteristics of the data. So when designing an fMRI experiment, you have to balance the need for adequate spatial resolution with that of adequate temporal resolution. So the temporal resolution determines our ability to separate brain events in time. In fMRI, the temporal resolution is determined by how quickly each individual image is acquired. And this is determined by something called the TR. We'll return to this as we move along. The spatial resolution determines our ability to distinguish changes in an image across different spatial locations. So, for example, when we have a structural image, these are often called T1 images, and again, we'll return to this in a later module. These have high spatial resolution, but low temporal resolution, actually no temporal resolution because they're just a static image. But since they have such high spatial resolution, they can be used to distinguish between different tissue types. And so we see this cartoon image here that you can separate between gray and white matter, and you can make out anatomical boundaries and whatnot. Functional images, which are also called T2 star weighted images, and we'll talk about that again as we move along, have lower spatial resolution, so they're much blurrier than their structural counterparts. However, we can measure many of them, and so they have higher temporal resolution. And they can therefore be used to relate changes in signal to an experimental manipulation. So in this cartoon here, we have a bunch of images that are acquired while you're performing condition A, and a bunch that are performed while you're doing condition B. So in our last module, we gave this little example where you were finger tapping, which might be condition A, and then you're resting, which could be condition B. So you have measurements under both conditions, and then we might want to look at the differences in signal between these two different conditions. And that's something that we can do with functional images. Another piece of terminology that's important is different types of slices of the brain. So we measure a three-dimensional brain volume, but we often study slices of the brain. So we often talk about things like coronal slices, which is a slice in this direction, sagittal slices, which is a slice in this direction, and axial slices, which is a slice in this direction. So these are examples of coronal, sagittal, and axial slices. fMRI or MRI images are typically acquired in axial slices one at a time. So we go and do different slices in the XY plane like this. They can be performed either sequentially, so one slice at a time, or interleaved, where we skip a slice and then come back and do it again. We'll talk about this later on. Together, these slices are then kind of glued together and made to make up a three-dimensional brain volume, which is what we ultimately analyze. There's a bunch of terminology that we need to describe the acquisition. The first thing that's important to know is what's called the field of view. The field of view is the extent of the brain that's inside of the image. So for example, the field of view might be around 20 centimeters. In this example, I have it at 192 millimeters, so it's 19.2 centimeters. So that tells us what's the extent in each direction of the brain volume. The brain volume, again, is made up of multiple slices, and each slice has a certain thickness. So we also have to say, what's the slice thickness? Here, an example, we might choose a 3 millimeter thick slice. Each slice is then split up into voxels. And for example, in this cartoon, the voxel, we have the actual slice is split up into 4,096 different voxels in a 64 by 64 grid. So if we have 64 voxels and our field of view is 192 millimeter, then each voxel is going to be 3 millimeter in both the X, Y, and Y directions. So in this case, if we have a matrix size of 64, a field of view of 192, and a slice thickness of 3 millimeters, our eventual voxel size will be 3 millimeters by 3 millimeters by 3 millimeters. And so that's going to be the size of the unit of measurement that we're going to be interested in fMRI. We can change that. For example, we can make the slice thickness thinner or thicker, or we can make the matrix size higher and thus make the voxels smaller or bigger in the XY directions. So An experiment studies many different subjects. So typically in fMRI, we have experiments where we look at multiple subjects that perform the same type of task. And each of these subjects might be scanned during multiple sessions. Each of these sessions consists of several runs. We might repeat the task several times on each subject while they're in the scanner. And each run is going to consist of a series of brain volumes. Now, each of these brain volumes, again, is made up of multiple axial slices, and each of these slices contains many, many voxels. And again, each voxel has an intensity associated with it, and this is the basic measurement in fMRI. So this sort of suggests that there's a hierarchy going from the experiment, there's subjects nested within this experiment, there's sessions nested within each subject, there's run within each session, and then there's brain volumes within each run. Each brain volume consists of are measured sequentially over time, and each voxel has a corresponding time series associated with it. So this is basically the final level where we analyze the data on this time series level. But again, we also want to be able to look at these time series and say, what can they say about the population of subjects that we studied and whatnot? Okay, so that's the end of this module. Here I've just attempted to kind of talk about some characteristics of fMRI data, talk about some nomenclature that's going to be important, some terminology, and that will help you read method sections and whatnot. Okay, I'll see you in the next lecture.